So welcome back, everyone, to the last session of this symposium on how microbes rule the world. And the world is not just oceans. Surprise. So to help us realize that, next speaker is talking about terrestrial environments. And this is Francis Martin from the French National Institute of Agricultural Research. Very welcome. You are a high-level researcher on fungi and symbiosis and terrestrial microbial ecology. So very welcome. Talking about the evolution of modes of nutrition in forest fungi from wood decay to symbiosis.
Thank you. Thank you. Well, I'm like an outsider today, but still I really enjoyed my day. I mean, all the speakers today were outstanding, and I'm very pleased to be there. And I've always followed marine ecology because oceans are more simpler ecosystems than so. and so you are really at the cutting edge of ecology sciences. So I will try to bring you to the woods today as a change. So my duty in life currently is to save the trees, like these sequoia trees which are currently dying in California as a result of climate change. And I think that one way to save the trees is to use microbes and to get a better understanding of the role of microbes, including fungi, in forest ecosystems, in the functioning of these forest ecosystems. So today I'm going to briefly summarize the research we do in my department and within a large consortium of people working on forest ecosystems and the role of microbes in these forest ecosystems. And I'm going to take one example of how we can use genomics and more specifically comparative genomics to understand, to harness comparative genomics to understand microbial ecology in forest ecosystems. And I'm going to talk about the evolution of the mode of nutrition of a group of fungi living in forest ecosystems. I'm going to show you that these fungi are main actors in the carbon cycling in forest ecosystems, both as decomposers, but also as symbionts. And many of these fungi, symbiotic fungi, I'm sure you know them very well. I'm in Sweden there, and a lot of people in Sweden are eating mushrooms. And so the fungi I'm working with are called chanchells. You know them? Porcini, morels. So you see, being a fungal microbiologist has a lot of advantage. So we can go to the woods and collect mushrooms. But it would be good to have the screen right now, because I'm not going to entertain you. Of course, I can talk about mushrooms for hours. If you want to know everything about the sex of the black truffles of Périgord, I can talk about that, but I'm not sure that the committee would be pleased. Yeah, anyway, we have to use... You can see? Okay, so I was ready to tell you that I'm going to introduce and to present you what are the mycorrhizal symbiosis. Then I will talk about one large-scale program called the 1000 Fungal Genome Program, where we are aiming to sequence the genomes of many environmentally relevant forest fungi. And then I will show you a few data on the evolution of these forest fungi. And finally, I'm going to briefly summarize 200 million years of fungal history in one slide. And if I have time, but time is running short, you are not counting my time, right? No, okay. Then I will show you how these symbions are talking together. We've been able to identify a few signals and molecules which are involved in the dialogue, the molecular dialogue between the two partners. So, again, a bad joke. You believe that this planet is a blue planet. I think it's also a green planet, because 1/3 of the continent, 1/3 of the land is covered by forest ecosystems. Of course, you know that forests are very important in the northern part of Eurasia and Canada, but also the Amazonia and the Indonesia. And so people working on these forest ecosystems, they are starting to know a lot on the interaction between the plant species, the tree species, both in the boreal forest, where there are a limited number of tree species, but also in subtropical and tropical forests like this Yunnan forest, where there are more than 500 species of plants and trees interacting. But there is one thing that people know very little about, the role of microbes. I really need the right picture there, right? If not, you won't be able to see the tight interactions between the root systems. So we try to understand the role of the microorganisms, and more specifically the fungi, in the tree and forest ecology, and how this fungal hyphae, the fungal network, the fungal web, are connecting not only the trees belonging to the same species, but also linking and bridging the trees belonging to different species. And sometimes we know that, for example, that orchids could also tap this fungal network connecting the different plants. So you should, next time you walk in the woods, just think, not only look at the beautiful organisms that the trees are, but also look at your feet and think that below your feet, when you walk, there are webs of hyphae connecting the plants. And through this hyphae, we know that there are carbon, but also signals moving from one plant to another one. So we try to understand the role of these microbes in the forest ecosystems. So the main goal of our research program is to understand the plant-microbe interfaces. For us people working on forest ecosystems, understanding the interaction, the interfaces between fungi, like mushrooms, and trees are crucial, because these fungi in forest ecosystems, they play a key role in carbon cycling, carbon sequestration. So our goal is to characterize these interfaces between trees and microbes, which are allowing the transfer not only for energy of carbon between the different components of the forest ecosystems, but we believe they are also involved in the transfer of information and different kinds of signals between trees and their environments. And of course, by looking at this slide, you are, I hope, realizing that I'm very mycocentric. The mushroom is bigger than the trees. Because currently, at least over the last 10 years, of course I'm a plant physiologist by training, but I realize that we cannot understand the biology and physiology of trees without understanding the role of these mushrooms. So currently we try to define the fungal communities in forest ecosystems. So this means large-scale metabarcoding to identify the different species and ecotypes of fungi living in the forest. We want to characterize the interactions between these fungi and plants, but also bacteria and protists. And we try on a few sites to manipulate these interactions by changing the environments, rain and water and heat, sometimes the soil, to look at the impact of these changes in the environment on the interactions between fungi and trees. And we do all this work because we hope that we can feed the models. Currently, most people working on forest ecosystems and most terrestrial ecosystems, they don't mind about microbes. They want to forecast the impact of climate change on forest ecosystems. They want to forecast the carbon cycling in these ecosystems. But for them, microbes is just a black box. They don't know how these microbes change over time and under the impact of the climate change. So we hope that this data that we are gathering today will be used by modelers to forecast how forest ecosystems will behave in the next 5 to 50 years. So let me introduce you to the main actors, the main fungal actors in a temperate forest or a boreal forest like the forest around Lund. There are let's say 3 main groups of fungi in forest: the decomposers, the decayers, the symbionts, and the pathogens, the parasites. So regarding saprotrophic species, we have the white rotus, the one able to decay wood, to eat both cellulose and lignin. We have the brown rotus, able to make their way through lignin to access cellulose and to degrade cellulose and to use glucose. And so these 2 guys are very important. This is the ones which are decomposing wood. They are amongst the only organisms on our planet able to decompose, degrade wood. So they play, of course, a major role in carbon cycling. The soil saprotrophs, they are able to decay organic matter, twigs, dead leaves, but they are less able to decay and decompose wood material. And then these are my favorite one, the symbiotic species. They have learned how to talk to the trees through the last 200 million years, probably more, and they are fed by the tree in carbohydrates. So they set up a symbiosis with trees. They are providing them in mineral elements and water, and as a reward, they are getting the carbon. And then we have the pathogens, and as a forester, I don't like them so much because they are able to kill the tree to get access to the organic matter. So we want to understand the role of each of this group of fungi, but I'm mainly focusing, we are really focusing on the symbiotic fungi able to form an interaction, a mutualistic interaction between trees and fungi called ectomycorrhizae. Mycorrhizae mean roots and fungi together. They are forming a symbiosis, and as you see there, many of them are quite edible. I don't recommend to eat that one, but at least we have porcini, matsutake, yellow chanterelle, black truffles. These fungi, these are the mushrooms of the fungal net colonizing the soil and colonizing the roots and forming this kind of structure where you can recognize the central part of the roots, the cortex of the roots, and the root is ensheathed, enclosed by aggregates of hyphae colonizing the roots. And these hyphae are able to extend inward between the root cells to form the symbiotic interface where all the exchanges are taking place. And they're also able, you don't see that on that slide, but they're able to extend the hyphae outside to explore the soil. So these fungi, we know at least 20,000 species of symbiotic mushrooms. Their ecological relevance is very high. I hope I've been able to convince you about that. Many of them are edible, and the genome size of these eukaryotes is smaller than plants. It's between 20 megabase and 200 megabase. So they are very, sequencing centers are very keen to sequence them. So what the role of this interaction is mainly to provide nutrients and food to the plants. And you see why? Look, each root, this is a pine tree with the root system there, and each rootlet is ensheathed by the fungal mycelium, forming white spots. And this is a close-up of these rootlets transformed by the interaction with the plant, with the fungus. But you see from this mycorrhizae, you have an extensive net of hyphae colonizing the roots, the roots, but also mainly the soil. And for 1 meter of roots, you have 1 kilometer of hyphae. So this symbiosis is increasing by a factor of 1,000 the uptake of nutrients. So that's the main impact of this fungal association, increasing plant growth and stimulating plant growth. And as a reward, the plant is providing carbon to the fungal networks. And we know very little about how this interaction is coordinated. What are the metabolic pathways which are regulated by the interactions? What are the signals? What are the mechanisms coordinating the activity, the metabolic activity between the plant and the fungus, but also, as you see there, between the different compartments of this complex interaction. So currently people are trying to fish out the genes and the protein and the signals which are involved in the coordination and the activity of this symbiosis. And that's mainly, again, because they play a key role in the carbon cycling in forest ecosystems. For many years, people thought that the main actors in the organic matter decomposition in forest ecosystem were these saprotrophic species like this one. And more recently, people like Anders Thunlid here in Lund have demonstrated that some of the symbiotic fungi may play a role in the decomposition of the organic matter. They may be able to get carbon from the plant directly, but in some specific settings, they can also decay partly organic matter. So that's one of the hot question currently. What are the role of this mycorrhizae in the organic matter decomposition? And to do that, we are using large-scale genomics to tackle that question. And we have 3 main aims. We try to relate genome features to ecological traits, and they are more specifically in organic matter decomposition. In other words, can you correlate gene repertoires to ecological traits? We also want to understand how these forest fungi evolved. Are the symbiotic species emerged from the wood decomposers and the soil saprotroph? And finally, we want to characterize the genes which are involved in this interaction, the symbiosis genes. I'm afraid today that I can only provide you some data on AM1 and 1 slide on AM2. So how did we handle that? It's very similar to what we've seen already today. We are using omics, genomics, transcriptomics, metabolomics, and all these large-scale, expensive tools to try to understand the evolution of these fungi and the functioning of mycorrhizae and the wood decayer. But first, as usual, if you want to get nice results, you have to sit and first you need to have a good sampling of your organisms, bacteria, or in that case, fungi. So we've been through the tree of life of fungi, mainly Basidiomycota, and we did hopefully a phylogenetic and ecologically relevant sampling of these taxa and genomes. And then we are using these genomes to look at the evolution of ecological traits, as I mentioned, but also we also improve the phylogenetic knowledge on the taxonomy and systematic on these organisms. And we can use this reference genome also to look at the symbiosis genes and do RNA or transcript profiling to identify, fish out the symbiotic genes. So the reference genomes are used by different people in the team from evolutionary biology to molecular biology. And we have been very lucky to collaborate with the Joint Genome Institute in California. The JGI is, as you probably know, focusing on the sequencing of organisms playing a key role in the environment. And we have been collaborating tightly with Igor Grigoriev, who is leading the fungal genome program at the JGI, and also Joyce Patafora, Dave Ibbett. And we have been lucky enough to get funded for two large-scale programs, the Mycorrhizal Genomics Initiative, where we are sequencing mainly symbiotic species, and more recently, the 1000 Fungal Genome Project, where we are sequencing 1000 genomes. So we started by sequencing this mushroom about 10 years ago, but now we have more than 300 species of forest fungi, saprophytic species, endophytic species, and symbiotic species. And we try to make sense of all this information. And to do that, of course, you need good bioinformaticians and good ecologists. So that's one of the first glimpses of these programs. So there you have about 120 genomes from ectomycorrhizal symbionts, saprotroph, pathogens, arbuscular mycorrhizae. I mentioned them briefly. I will get back to them later. And we have also wood decayers. And you see that the genome size is highly variable between the different groups. Most of the saprophytic species here, wood decayers and soil saprotroph, the average genome size is about 20 megabase. And so it came like a surprise when we got the first series of symbiotic genomes, because usually on the average their size is much bigger. Some of these genomes, like the black truffle of Périgord, the genome size is more than 200 megabase, so about 10 times larger than the average saprotroph. The arbuscular mycorrhizae, there are some of these arbuscular mycorrhizae forming symbiosis with most of the land plants are above 200. So we don't understand why these genomes are so big. Instead, except that we found that when we compare the genome size there between these symbiotic species and the rest of the sequence fungi, we realize that very often we can explain the size of the genome by the content in transposable elements. Most of these biotrophic species, most of these mycorrhizal species are overloaded by transposable elements, and we don't know why. And we don't know the impact of these transposable elements on the genome features, but also on the functioning of these organisms. So it's really an open field for the new generation. Look at the impact of transposable elements on the symbiosis functioning. So, this being said, we mainly focused, having all these genomes in our hands, we mainly focused on trying to correlate the features of the gene repertoires, like occurrences of the genes, but also gene acquisition, gene decay, gene duplication, expansion or contraction of gene families. We tried to correlate that information to the ecology of the ecological traits. And so whether the fungus is an endophyte or a symbiotic mycorrhizal species, or a leaf decayer, or rotor. And we mainly focused on one group of genes, those coding for polysaccharide degradation, carbon degradation. And just one slide to remind you that wood and organic matter in forest ecosystem is mainly composed of two biopolymers, very complex biopolymers, cellulose and lignin. So lignin and cellulose are making fibers, intermingled fibers, to make wood. And there are very few organisms able to decompose lignin. It's a very resilient biopolymer, and not so many organisms able to degrade cellulose. To degrade cellulose, you need endocellulase, exocellulase, and then the product of this hydrolytic activity, the oligosaccharides, are then degraded by a series of endoglucanases or glycosides. So if you code these genes in your genome, then you can decay cellulose. Lignin decomposition is mainly relying on oxidoreduction mechanisms, and fungi are especially Basidiomycota, are very good at decomposing lignin because they have peroxidase, including a class 2 peroxidase, a lignin peroxidase, which is able to degrade phenolic compounds of lignin. So all these enzymes, all the genes coding for these enzymes, are called K-zymes, for carbohydrate active enzymes, and many of them are plant cell wall degrading enzymes. And we are lucky enough in France to have the world experts in K-zymes. Bernard Henrissat in Marseille has set up a database where all the genes coding for this enzyme for any organisms or any metagenomes has been stored and annotated. So we collaborated with Bernard Henrissat to scan, to survey all these genomes of fungal organisms. And that's what we obtain. It's a complex site, so I'm going to go through quietly. So here you have 122 species of forest fungi, including brown rotors, white rotors, so wood decayers, soil saprotrophs, endophytes, and symbionts. So this is a species name. Don't try to read them. There's a few photos there, so you can recognize many of the common wood decayers, but also Coprinus and Lepiota that you can collect in pastures, so soil saprotroph. And there are a few representative of the symbiotic species. And here you have the number of genes encoded in the genome of this species, the number of genes coding for 9 of the most important K-zymes, the 9 enzymes you need if you want to decompose wood or organic matter. And so this includes endocellulose, exocellulose, but also lignin peroxidase. And you see that the number of genes coding for these enzymes is very different between the different species. Here, the ones having the largest amount of genes are coding for white rotors and soil saprotroph, so they are very efficient at decomposing organic matter. And then, on the right of the slide, you have mainly symbiotic species. So you see from, and there's a kind of break there between the soil saprotroph and the wood decayers and the symbionts. So the main conclusion of this large-scale comparison is that the symbionts have lost most of the genes coding for wood or organic matter decomposition, lignin and cellulose. So a few takeaway points there at this stage. So all of the sequence ectomycorrhizal species have a reduced complement of genes encoding plant cell wall degrading enzymes. And so this means that since they are not able to use organic matter from soil, they fully rely on the plant for the carbohydrates. They have no other choice than talking to the plant and getting carbon from the plant. But we see that it's not a black and white. There's a continuum of K-enzyme contents. So some of these ectomycorrhizal symbiont may be involved in some cases to partly decompose organic matter. So let's now have only 8 minutes.
Yes.
I'm going to summarize in one slide 200 million years of evolution. Whew. So by using these genomes and looking at the loss of genes, like those coding for plant cell wall degrading enzyme, we've been able to, using the proper algorithms, to reconstruct, by using phylogenomics, the history of this fungal species. And then we discovered that everything started about 280 million years ago, when one fungus was able to invent the lignin peroxidase, the enzyme of peroxidase of class 2, which allow these fungi to decompose lignin, the phenolic compounds of lignin. So these white rotors gave rise to many species, including, of course, a large radiation of white rotors. But then a few of them lost their ability to decay lignin and cellulose. They start to talk to the plant thanks to some signals and small proteins, and many of them are now symbiotic species. So this is one path of evolution, from the wood decayers, wood decomposers, to wood decomposers, and then to ectomycorrhizal symbionts. Probably the most common pathway evolutionary scenario is this one, where the white rotors gave rise to brown rotors, fungi not able to, they are able to make their way through the lignin, but not really to decompose lignin. They are mainly decomposing cellulose by using oxidoreduction mechanisms. And many of these brown rotors gave rise to the large family of Boletales, the Porcini, the Bolets. And now many of them are very abundant in the conifer forest ecosystems. And then the third pathway, leading to mycorrhizae, to symbiosis, wood decayers gave rise to soil saprotroph, like this Amanita, or like Coprinus, or Agaricus. And then some of these guys lose their ability to decompose organic matter, and then there are no symbionts like the Amanita. So it's a nice example of convergent evolution, and very special one, because in that case, this adaptation to the interaction with trees took place by loss of genes. It's very rare in plant systems that adaptation to a new environment is taking place through lack and loss of genes. So that's a good example of that type of mechanisms. Now, again, in only one slide, I would like to summarize what we have showed by using large-scale genomics and molecular biology, what's taking place there at the very early step of the interaction between the fungal hyphae, there in green, interacting with the short roots emerging from the main roots there in red. So what are the mechanisms which allow the fungus to interact with the plant? So we have been focusing mainly on a group of small secreted proteins. They are the red balls, which are released by the hyphae, which is there landing on the surface of the root cells. And we showed that many of these small proteins are used to glue, to anchor the fungal hyphae on the root surface, but some of them are able to enter the root cells, move to the nuclei in less than 10 minutes, and then they do interact with transcription factors in the root cell, where they control the plant immunity and the plant defense reaction. So I don't want to go in detail in that slide, but just keep in mind that this fungus, when it's interacting with the root surface, is releasing a large, at least 50 different small proteins, and we found that some of them are changing the surface of the plant cell, but quite a few are, again, penetrating into the root cells, into the plant nuclei, where they interact and change the plant gene expression. So the evolution of this group of fungi took place by at least two strikingly different mechanisms. First, by likely, this is my view, first the fungus needs to talk and communicate with the plants, and he does this communication by using small secreted proteins, effector proteins, injecting them to the plant cells to control the metabolism of the plant and to control the plant defense reactions. And then, when this dialogue is on, then the plant is providing carbon to the fungus, and then it can lose the plant cell wall degrading enzyme. The fungi, these fungi, they do not need this plant cell wall degrading enzyme anymore because they are fed by the plants in carbon. So that the main results, and we demonstrated that this evolutionary scenario is taking place in Basidiomycota, in Ascomycota, and more recently we also showed that exactly the same mechanisms is taking place in Glomerulomycota. The Glomerulomycota are also called arbuscular mycorrhizae. It's a very old symbiosis which probably evolved 400 million years ago between land plants and this very primitive group of fungi. And again, these fungi evolved very similarly. They have lost their plant cell wall degrading enzymes, and they are using small secreted proteins to talk to the plants. So over more than 400 million years ago, in most of the fungal lineages, the same mechanisms independently evolved. Next step, final slide, of course, we want to do as good as you did, you marine scientists. You are able now, and we have seen many examples today, you are able to look at the function expressed in situ in the ecosystem. So we are far away from that stage because we were missing reference genomes, like you mentioned earlier today. Now that we have 1,000 genomes of fungi, it's getting better. We can go to the woods, we can collect mycelium, we can extract eukaryotic RNA, we can sequence them, and we can map them to reference genomes to look at the function expressed in the soil, in the forest ecosystems, where this kind of symbiosis are taking place. So we are really at this stage today where we can, when we do sequence RNA extract from soils, about 45% of the reads are mapping to reference genome. So there's still some way to go, but it's getting much better. And of course, I would like to thank all my collaborators there. It's a symbiosis. All these people are working together, and it's really a crosstalk, a daily crosstalk between PhD students, postdocs, and staff scientists, and many collaborators there throughout the world. And now I'm done, but if you wish, I can share a few of my mushroom spots. I can talk about golden chanterelle recipes or the sex of the black truffle. Oof.
Thanks a lot. Do we have questions? Yeah.
Oh, that was a great talk. Thanks. I was curious, do you see any evidence for horizontal gene transfer between the fungi and the plants?
Yeah, I mean, that's really one of the main questions in the community. We tend to think that there is no or very little horizontal gene transfer in fungi. There's just a dozen of examples. So it's in contrast to bacterial communities, horizontal gene transfer is probably not one of the main driver of gene evolution in fungal communities. But you know, there's quite a few examples, but not so many. So I would say no. You mean exchange between fungi and plants? Since people are using RNAi and small RNA to transform plants, this means that fungi can exchange RNAi or small RNA with plant, and there is quite a few. Yeah, there is examples of that. So we believe that there are RNA transfer between. We know I showed you that there are translocation of small proteins between the fungus and the plants, and we believe that there are also RNA moving from the fungal side to the plant side. But there's only one or two groups working on that, so we are missing data.
Ramonas?
Very, very fascinating talk and contrast to marine systems. And I'm fascinated by the fact that this really complex, difficult process of degrading lignocellulose was invented a few 100 million years ago, and then the main direction of evolution was to actually lose it. Is that true, or is there, in parallel, the actual degraders of wood have been still co-evolving with the trees to trying to beat each other by building more complex lignin and then figuring out on the fungi side by how to deal with that? Or do you think the battle was over pretty soon after the invention of lignin?
Maybe Anders could help me there, but I think lignin has been invented probably 500 million years ago, 400 million years ago. We believe that the lignin or lignin-like components between 400 million years ago and 300 it was somewhat different from the lignin trees have now. But whether lignin is still evolving, I should confess, I don't know. I don't know. I mean, I think the fungi degrading lignin is good for the trees also, because in fact it's the main system to recycle nutrients. So you should not see lignin degradation as a drawback to the system. I'm really convinced that these wood decayers play probably the major role in ecosystems. Wood should be degraded. Some people think that before fungi invented lignin degradation, lignocellulose degradation, wood was accumulating on the soil and then quickly transformed in peat and coal. Until one day, this ancestral fungus invented class 2 peroxidase. And then the accumulation of wood on our planet stopped. And so people are arguing that then the coal accumulation stopped. Mm-hmm. So they are the main drivers of carbon cycling. Without them, without these white rotors and brown rotors, we would be just overwhelmed by detritus.
Thank you. Another one over there.
So your focus has been on fungi, but are there bacterial decomposers of wood as well?
5 years ago, I would have said no. But now there are people working on bacteria showing that some groups are able to decay and decompose lignin and cellulose. but at a much lower rates compared to this Basidiomycota. But there are a few bacterial systems in the gut of termites, in the gut of insects, beetles. Clearly, they are able to decompose lignin and cellulose. But as a fungal biologist, I tend to think that these agaricomycetes or Basidiomycota are the main ones, the stronger ones.
This will be the last question, Corinne.
Thank you. I might have misunderstood this, but to me it sounds like from your explanation that the fungi is really driving the symbiosis, because it's the fungus that invades through the root and takes control of the plant. Is that correct, or is there a crosstalk?
This is a current speculation. This is a current hypothesis. But keep in mind that the plant is still playing a key role, because the plant is providing glucose to the fungus. So the fungus cannot survive without the plant carbon. And more interestingly, we have one project running now where we show that the plant, when I say the plant for us is the poplar tree, which is the main model system, poplar is also injecting small secreted protein in the nucleus of the fungal model. So what I showed you for the translocation of small secreted protein, effector-like proteins from the fungal component to the plant component, the tree is probably also injecting the same kind of molecules and proteins in the poplar, in the fungal cell. So it's really what I like to use a crosstalk. It's really a crosstalk. Each one is controlling the other one. Thank you.
Thanks a lot.